Good morning, everybody. We're going to get started in just a second here. So this is the Data Standardization, Normalization, and Integration session. I'm Dan Gilchrist. I'm a program officer with the NIH at National Human Genome Research Institute. I work on functional genomics and informatics, and I help manage the ENCODE project as well as the HubMap project.
Good morning, everyone. So I'm Caroline Uhler. I'm a trained statistician, now a faculty at MIT in electrical engineering and computer science. I'm particularly interested in problems relating to the codes of how genome architecture and gene regulation, gene expression are linked to each other.
I'm Peter Kurchenko from Harvard Medical School, and we've been working for some time on developing computational methods for genomic assays, in particular single-cell assays. And in terms of, I think, biology, we've been applying it to studies of different types of cancer and also development and normal tissues such as brain and so forth. And I guess the main topics of normalization, integration, and standardization, they come up in pretty much every project, and obviously in HC as well. You heard some of this yesterday. So this is meant to be, I think, a more interactive session. Yeah, I'll walk over there and talk about the slides. So we prepared this initial list of questions, and I think everyone has a printout of this. But realistically, so the kind of among the three topics, the key points that we thought were important or potentially important to discuss were the following, as just a quick summary. And so I'll go through them in a second, but what we thought we would do is actually break you up into smaller sections initially for, I don't know, about 20 minutes to try to come up with a list of questions that seems to originate, let's say, from your experience, from actual problems that you're facing in the lab. Because I think for a lot of us, these seem to be very top-level questions. Maybe there are more specific things that could be addressed. And then after this little breakout period, we'll basically try to collate the questions, and we'll ask you to discuss some of these. And hopefully that will lead to kind of a productive discussion. All right, so just quickly, I think our thoughts, and please feel free to chime in on this. So I think for standardization in particular, the question of quality control metrics comes up all the time. There are some guidelines that are obviously built into the pipelines that exist, but as far as kind of standards, it's really, I think, undefined. This also comes up a lot, when you start comparing data from different teams that are using different protocols. We know there are systematic biases, but can we come up, let's say, with quality standards that would be acceptable for both techniques like SmartSeq2 that has very high coverage, relatively speaking, as well as something like MicroWell that tries to measure a lot more cells, but at a lower coverage. Data normalization, so you heard from Dana yesterday an example, for instance, and I think there are quite a few methods that have been proposed for normalizing particularly transcriptome data. One of the questions we thought is whether this should be formulated in the context of specific analysis or sort of general kind of general corrections. So let's say if you're trying to run differential expression between two clusters, right? Should you go back and reconsider normalization in this context, or should you try to come up with a method that will sort of normalize it first and then use it for everything else? And related question for normalization, obviously, is how do we make sure that we're not over-normalizing or over-correcting and so forth? I guess an obvious example of this would be like regressing out cell cycle together with differentiation or something else. The last topic is, I guess, more popular now, and within our group had most enthusiasm, is data integration. And there are quite a few methods that have been presented, and particularly if you follow bioRxiv, you'll see a variety of things that are coming up. But they're also addressing different problems, it seems. Some of them are looking at more or less technical batch correction, others are trying to integrate across, let's say, different individuals, different tissues, or different species maybe, or different modalities, molecular modalities. So, key question we thought was, from your standpoint, what are the types of data that you feel needs to be integrated that is objectively needed in your studies. What are the sort of scenarios? And we think, especially in the computational community, it's kind of attractive to think of integration problem as one problem, and that you integrate once and then you use this result for all kinds of downstream analysis. But I think it's important to consider what the downstream aims are. And so I think formulating this again to get your perspective on that would be quite useful. And finally, within this context, how do you think quality of the integration can or should be assessed? Can we come up with controls, some specific experimental controls that would work for this, or general computational metrics? Anything you want to add to? Yeah, do you want?
Okay, so that was just a couple of ideas that we came up with, but I think the goal would be for everyone here to think about what are the main questions that you think are unsolved with respect to data standardization, data normalization, and data integration. And so since we have 1.5 hours, we thought we really want to go a bit deeper and get to concrete problems that you think are important. And so we were thinking of first starting off with actually discussions in smaller groups, say, I don't know, about 5 people or so. And I think it would be great, and for this we would like to get a bit of a feeling for who the people are, who we have here, like how many are experimental, how many are computational, so that maybe we can actually have a bit of a more mixed group. So maybe you can just also look around and maybe we'll just do a show of hands of like who considers themselves mainly experimental. Okay, everyone. So who considers themselves, oh, okay. And who considers themselves mainly computational? Everyone else. Okay, so that's good for us to know. Okay, so they won't be very mixed groups, but I think it would still be great if we can actually have some smaller groups formed by, however you're sitting, or it would particularly be great if you actually discuss with people you've never talked to before. And think about the 2, say, main questions that you think are unsolved for each one of the 3 topics. And it would be great if you can be as specific as possible so that we can then, really try to then in the next step kind of prioritize some of these questions and then actually go more into detail for these questions. So we're thinking of about 20 minutes for just discussing in the smaller groups so that you also have time to introduce yourself and then really talk about the main questions that you think. So I know this room is a bit not ideal for this, but maybe you can self-organize into smaller groups. And we'll just be going around and being part of some groups as well. Okay, so let's get started.
Okay, everyone. Thank you for taking time to discuss it. So we put together a quick list, and I'll try to project it in a second. That essentially groups the topics that you've been covering and lists different teams for each question. Let's see if I can project that. Can I get the HDMI channel, please? Thank you.
桜井さん、お差し上げます。はい。どうですか？ OK ですか？はい。うん。Yes. はい。
Excellent. Thank you very much. Okay, so basically you'll see group numbers here next to each question, and we'll just try to lead in an order. And the idea is to express your thoughts, ideas, and we'll try to record it and then summarize. So the first topic will be QC, and there are a few groups that basically try to separate sample level QC and cell level QC. So I would ask, group 4 and 2, for instance, can start, and then there will be a subtopic of spike-in, where group 5 and 1 wanted to say something. So some representatives of the group should come up and just express their thoughts, I hope. So anybody from group 4? I didn't warn you about this, did I? Just summarize, quickly the thoughts that you have.
Okay, so group 4. So we discussed sample level QC and cell level QC separately. For sample level QC, we talked about running basic analyses like fast QC and getting an idea what the quality of the input data is, getting a % mitochondrial read overall, and when available, getting approach-specific data. For example, for the C1, we can get images of cells and get a quality score on that. We also talked about getting an overall percentage of spike-ins and what other metrics we could use. It's like an open question.
Mm-hmm. And then let's say for spike-ins, because I know some of this discussion took place, did you have suggestions? Because obviously a lot of groups already have experience, let's say with the RCC spike-ins and so on. Did you feel like more effort is needed or in different direction? Or?
Well, what we wanted to do is for spike-in, we want to have an idea of what % of the library is a spike-in. So we can identify empty wells, for example, in Smart C2. Nothing beyond that.
Mm-hmm. Okay, group 2. Yeah, I think we have a bunch of mics somewhere. But yeah, there's another mic there.
Right. So yeah, our question here was whether there's a way to avoid avoid thresholds when we do cell QC. So whether we can do this in an automatic way, preferably a subpopulation-aware way, where we just learn the thresholds from the data. So that was our question for the QC.
So the current methods, right? They do have some heuristic that they use to pick a threshold. So did you essentially want to make that better, smarter, or did you mean something else?
Yeah, so I think a lot of people in the group still are picking arbitrary thresholds for things like mitochondrial or number of genes. And in the same way that empty drop models the background, we'd like to be able to model the distribution of these statistics on many of these statistics and then learn what the normal distribution of them is, and maybe just do automatic outlier detection so that you don't need to consider the choice of statistic, and that should be aware of the structure of the data so that you have different thresholds for different subpopulations.
Okay, great. So I think groups 5 and 1 particularly talked about spike-ins. Did anybody want to add to the general desire to have spike-ins?
So from group 1, on the discussion of spike-ins and basically all of the early stage QC, we kind of all agreed it is super dependent on what assay you use and what the specific biology is. And so to the point of automating it, it's super tricky. Like maybe some of these later steps would be easier to automate, but these first few ones are so incredibly dependent on the biology. Right now, we were just like, yes, it's a very big problem.
Mm-hmm. Okay. Let's see.
We also discussed getting an idea of what kind of clusters we expect. So if, for example, we see one single uniform cluster in a sample, that would probably be a way of classifying bad samples unless we have information that a very uniform cell type was getting sequenced. Mm-hmm. So getting that type of information in would be useful to automate it.
Okay. Any other comments on cell level QC? I guess we discussed the thresholds already. Okay. All right. So the one of the sort of angle on this, I think, from Group 6 has been the extent to which these thresholds and QC metrics should be enforced by DCP. And does Group 6, did you want to explain the thinking? So I think it was over here, right? Sorry, it was Group 5? Okay, my bad.
Yeah, well, the thought was essentially whether there should be a minimal set of descriptions and in the data sets so that the Human Cell Atlas accepts that data set as part of the consortium, so that it becomes really useful for others to know what happened with that sample, how it arrived there in the metadata descriptions, rather than just letting users to submit whatever they want.
Mm-hmm. Any thoughts on this?
Thank you. I think this is a really good question. In other contexts, a similar question has come up. What kind of data can be submitted to something like the DCP or a data coordination center? And in those cases that I'm familiar with, the conclusion in the end was, well, we might want to submit data so we can share it with other people who might be developing an assay, and we don't even know yet what the right quality is. So the conclusion was essentially, well, we want to put, have clear QC statistics, but not prevent people from submitting their data. I think that's a decent trade-off. You just tell people what they can expect from your data.
Yeah, so as a member of the DCP, I can say I totally agree that we should have a way to capture things that quality metrics from the level of doing the experiment, not just metrics downstream after processing. So we do actually have a way to capture spiking controls at the moment, but I would caution that the DCP shouldn't be enforcing or setting those standards. I think it really should be the HCA community, like the much wider community, that decide maybe what's important, and also to prevent preventing people from being able to submit their data, because we do want to lower the barrier. I think that we can make these things optional or suggested, but maybe required, maybe too strict, because if someone doesn't do a spike in control, but it's a really valuable data set, we don't want to maybe prevent that. So those are my thoughts from the DCP.
I think the idea was more like, we have all these platforms, and I would just put the case, for example, of 10x Cell Ranger with this summary HTML file, which has a lot of information, very useful in terms of number of reads per cell, UMIs, all those kinds of things. And some other platforms that are more developed in the academic side, which I agree are completely useful as well. But I think there is a need for some sort of minimal way to compare two experiments. And some of those informations, the spikings probably are problematic if you didn't include them in your experiment, but some other metrics can be relatively easy be gathered from sequencing, any sequencing run. And yes, the community has to decide which of those metrics are the most useful.
All right. Okay, so if this is it for QC, let's just move on to normalization. We had a number of groups basically discussing this, and I think there are several kind of overlapping topics related to the generality of normalization, basically how widely it can and should be applied, and also I think the assumptions that are underlying a lot of these methods. So I think group 4 and group 2 has been discussing. So anybody from group 4 wants to express their thoughts?
I'm from group 4. We discussed the idea of normalization, basically making assumptions about something about biology. So even something as simple as library size normalization makes some assumption about the RNA content of a cell, for example. And so I think that, having a global normalization across something like the HCA is probably, you have to be very careful about doing that and careful about what you're trying to assume. One of the things we discussed at the end, after you came, was something I'd personally like to see is just like some sort of PCA or dimensionality reduction of the whole data set to understand what the largest sources of variation are. Is it tissue to tissue, really? I wouldn't be surprised if it's actually like platform, say SmartSeq2 versus 10x, which is also confounded usually with library size anyway. So to understand firstly what the largest sources of variation are, and then develop methods that try and reduce those types of variation. What we want to see is the tissue-to-tissue and cell-type variability. We don't want to be seeing all those sort of technical artifacts, I guess.
So if I get you correctly, let's say you have a process data set that has been normalized, and what you would like to see essentially is the picture of the aspects that were normalized out, like an overview, initial overview of all sources of variation before normalization?
I think initially you could do it without any normalization, and I'm guessing that the largest source of variation, it'll be pretty obvious, is either, well, actually I don't know, right? Because I haven't seen it, but is it just all the SmartSeq2 cells over here and all the 10x samples over here, potentially?
So you'd like to see that, you know, before normalization, essentially?
Yeah, exactly. Okay. And then we know that that's the biggest source of normalization. If you then go and normalize by library size and then look at it, do you still see, and I would think this is probably true as well, would we still see SmartSeq2 samples separating completely from 10x samples? And then we know that we have to build that into some sort of normalization. And then then do the tissues start falling out from each other, which is what we would hope happens, because they should be the largest sources of variation in the data set.
Okay. Group 2?
Yeah, so I don't think we have much more to add on that. I think there was some thought in the group that it would be good if you can sort of choose what normalization you apply on the DCP platform, whether it's into individual, batch, cell cycle, so that you can look at all of these different potential confounding sources of variation. Maybe I can speak to the next benchmarking point. So for this, we thought it would be good to have some sort of guidance on what type of normalization technique might be appropriate for your data, and for this, it could help to take perhaps some community data sets where effects are well understood. So there might be batch effects, they might be biological effects, and to really run the whole sort of gamut of available techniques against them and provide that as a resource for the community, so they can say, okay, my data type or scenario is similar to this one. We think that then this will have an expectation that they should perform well, and considered that how do you define success is obviously a question there, and one of the things that was discussed was maybe for differential gene expression.
So were you thinking that additional data sets are needed for benchmarking, you know, for different types of steps?
It's not clear that they are, I think, because there's already quite a lot out there. So there are, you know, the good examples of stimulation experiments where the biological effect is very clearly defined. There are good examples of batch effects. But yeah, it might be that you would want to design an experiment as well.
Anybody else wants to chime in on this part? We also have in the normalization, we had sort of a, well, let me, I summarized it, and now I'm not sure I summarized it correctly, but from group 3, it was a sort of a meaningful definition of a significant variation. I think they were discussing how to basically determine whether things, let's say you see a difference between two clusters, is this biologically meaningful or not, if I understood correctly. But group 3, can someone try to summarize this point if you understand what I meant?
Yeah, I think we were trying to get towards, like, when can you say that two things are statistically different in this case? Like when you're comparing gene expression, I guess that's sort of obvious. But if you're comparing cell type, when do you say that two things are different? Like, what's a meaningful difference? Say if you're comparing two patients, when is a cell different to another cell in that sense? Where are the boundaries?
So I'm just trying to figure out the contrast, let's say, with differential expression, right? So, or a clustering problem. So traditionally, you know, you see basically how well you can separate the cell in one cluster or another, or you once you have groups, you know, you test whether it's differential or not, differentially expressed or not, let's say, and you're trying to pick some other nuance here, right?
No, I think it's related to this. We ask questions like, if you cluster an organ, is the same level of variation that have the same meaning for different cell types? So if you see the same difference between, I don't know, 2 clusters of T cells or 2 clusters of hepatocytes, does that have the same biological meaning?
Mm-hmm. So let's say amount of variation expected from different cell types or in different tissues, right? All right, and then, yep, please.
Sorry, I was just wondering if I could clarify this point that I've seen some people working with this question of, instead of looking at the bulk data and saying what is the difference in the means, what's the significant difference, instead looking at, is there a difference in the variability? So instead of seeing variation as this confounding, this noise, is that the signal? Can you say that this cluster is more variable and more heterogeneous than this other one? So there doesn't seem to be a very well-defined metric or approach to doing this. Some groups are working on it and it's in progress. But maybe, perhaps, is that what you were getting at?
So is this a matter of, you think of having appropriate benchmarks for it, appropriate examples to be able to quantify this, to refine the methods?
I think what we need is more of like a statistical test or a new threshold for what does differential variability mean.
Mm-hmm. Yep. And some of this has been done, I guess, in the context of specific studies, like John Marioni, I think, published a year or so ago on mouse aging, and they were focusing on this question. But yeah, it's not very commonly used. The last point we had here is sort of, let's say, more forceful normalization, like taking out a cell cycle and so on. Well, how widely it should be applied and what sort of control should be given to individual users and so on. Please, group 4.
So yeah, from group 4, we want to understand what things would be useful for users to have removed from the raw data, and if they would want metrics, for example, where the cell cycle and or other effects like mitochondrial content was removed. And this also applies to a global background. So we do know that for data like 10x, there is some kind of global background from live cells. It would be useful to identify and remove that and provide these matrices to the user.
Any other thoughts on that? So methods for basically regressing out particular signals have been described in the past, obviously available, and they're used in some cases. But as far as I know, most of the default processing doesn't do this, right? So the question, I think, is whether as part of DCP processing or the way we present the data, there should be options to do these types of removals.
I think the amount of soup is fairly sample dependent. I think it depends on where the cells are from and which the organs are from. So I'm not sure that would be a globally applied thing. I don't know that we understand enough what the background looks like across all the samples.
Okay, let's move on to integration. So one topic that was sort of discussed, I guess, at different integration scenarios, so that I kind of grouped it this way. Various settings, let's say integration across platforms, integration across species, and so on. And a few groups discussed it. So we always start with group 4 because that's the first one I had here on the list. But how about we start with group 2? I think they wanted to discuss human versus mouse integration and simulations related to this.
Yeah, so the motivation here is for people working with mouse disease models, essentially, to understand how well they relate to humans and human disease, and that's the scenario. So just, are there better ways to be integrating between mouse and human than, for example, just clustering based on orthologous cluster markers between the species.
So do you feel like specific methods need to be developed, let's say, or adopted and for, let's say, just human-mouse integration, to have like a predefined set of genes, let's say, that seem to be trustworthy in terms of their behavior across species, something like that?
I mean, ideally, a method should go between any two species, right? I'm not sure it would be specific to mouse, because I mean, also if you're interested in developmental biology, then you would like the whole range of model organisms. So, yeah, it's not a very well-defined comment, but we would just like those kind of methods in a way that goes beyond what seems to be typical at the moment, which is just sort of comparison of cluster markers.
Mm-hmm. Okay. Other thoughts on, let's say, interspecies integration in particular? I like this topic. All right. Nick?
Yeah, I mean, we discussed to what extent this can be automated, and the conclusion was it's probably quite difficult. To do it in a completely unsupervised way and integrate data sets. Mm-hmm. Somebody will have to kind of manually look and make sure that the corrections we're applying are appropriate.
Okay.
Just to add to that, just from our group, I felt in particular that, again, it's what you're trying to do is going to determine which sort of methodology that you are doing. So if you're doing cross-species, you'll probably potentially have a different sort of tool that you're using to do integration than if you're trying to, integrate something at a finer level.
Okay. I think, let's just extend it to other integration scenarios. I think one of the things that was mentioned in group 6 was integration across platforms, and if we're assembling this collection as an atlas, right? How will we deal with adoption, let's say, of new techniques or improvements of existing ones, next versions, and so forth? Can we just do this using integration methods? So group 6, anybody wants to elaborate, please?
Yeah, so from group 6, we discussed about what new information each sequencing technology can add into the existing technology. For example, when we combine ataxic with the RNA-seq, do we add new regulatory information into the RNA-seq data? Or when we combine the RNA-seq with spatial technology, do we detect new cell type? Or can we reinforce the cell type that can be detected from both technologies? And then there will be challenges as well, like how do we normalize, or how do we integrate different data types that have very different distribution characteristics, for example, imaging data versus the sequencing read data.
Mm-hmm. And so for imaging data, do you primarily envision like you want to go beyond, let's say, spatial transcriptomics kind of things, right? That is still essentially measuring transcription. You're talking about morphology or other types of imaging, is that right? Okay. Let's see. Yeah, I think in this integration, we also lumped in the topic of, number of samples of sort of general experimental design that groups 3 and 1 has touched on. I guess, sorry, I can't spell number. I guess, if we're planning on relying on integration techniques as a way of, assembling an atlas, then we have to account for their limitations or abilities, right, in planning things. And I don't know, groups 1 or 3, do you want to elaborate on this a bit?
I think from us it was more like a precautionary thing where for a lot of us at the moment, like the cost is limiting and maybe we shouldn't rush to add lots of data if that's at the cost of good experimental design.
Mm-hmm. So cost considerations in the experimental. But what would you like to see? I mean, as a concrete, product or tool in this regard?
I mean, I think it sort of comes back to this being able to say if things are really different, right? I mean, if you don't have a good enough number of replicates, then can you really say that what you're seeing is true or not? So I think we were trying to say, like, not to rush to add lots of things, but to try and be very sure about what you're seeing first.
Mm-hmm. Is that fair? Don't rush with production.
And the one comment we had here was trying to think about what steps we can do in terms of protocol harmonization and experimental design alignment that with specific consideration of how it will then be integrated downstream, and what things we can do upfront to make sure that we're kind of like front-loading our ability to integrate them later.
Okay.
So there are a few specific suggestions, but I see that we're out of time.
Yeah, we're almost out of time, but in a sense, we're just going to stop and summarize for what we heard in the discussion and individual groups. So let's just finish this one thing: the Atlas-wide integration. I mean, to what extent people think that's even needed, and are there sort of scenarios where it will be useful, or the opposite? You should never try it.
So this was actually cross-Atlas integration.
Cross-Atlas,
yeah.
I'm sorry.
Yeah, and so, yeah, you should actually, you had the best.
Oh, was this, I don't know, there's been a lot of translation here, and some things might have gotten lost. So I think one question about cross-Atlas integration is to ensure that whatever HubMap does is compatible with what HCA does, which was like the lung map, et cetera, does. I think that is a really important integration question, because there's overlap, and if that overlap then means that these things completely disagree with each other, we certainly have a problem. And
okay, so you mean in more like coordination than integration, not like computation integration.
Coordination to enable integration.
Coordination to enable integration. Okay. Yeah. So obviously that we had this other topic that has to do more with standards, and that kind of goes into coordination between consortia as well. We'll try to reflect that. Okay, so I think we're out of time. I have 5 seconds, but thank you very much for spearheading these discussions and giving your input. So we'll try to put this together, and then please, during the main summary, if you feel like something needs to be added and clarified to the main group, then just come up to the mic and help us. Thank you.